Hello everyone, my name is Babak Saremi and I'm a PhD student from the University of Veterinary Medicine Hannover in Germany and work in the Department of Bioinformatics. Today I want to present you a slice of my work called Studying the Reproducibility of RNA-seq Experiments by Generation of Artificial Replicates, a Comparison Method. So our goal, as the title might suggest, is to study robustness and reproducibility in these experiments. And to do this kind of things, normally multiple sequencing experiments would be necessary, but this would also be very expensive. And that's why we try to study if we can use artificial samples to bypass the need and to increase robustness of RNA-seq experiments. And for this, we infected mice cell lines with the Batai virus and sequenced our samples in 2 batches to obtain sequencing data from 2 identical experiments. Here we call them R1 for replicate 1 and R2 for replicate 2. And each data set contains 5 infected samples and 4 control samples. And with the results, we did a differential. Here, I brought you the results of p-values and log2 fold changes of the differentially expressed genes from both of the experiments, and we can see here that both sequencing experiments have a strong correlation overall, but we still detect genes which are differentially expressed in one sample but not the other. And our aim now is to increase the robustness of our experiment by using artificial replicates. For this, we are using the principles of bootstrap method and draw reads from our FastQ file to generate new samples. So this is the path in the workflow right here. And we compare our method to 2 different common ones. One being mixed observations, where weighted mean is applied to the expression metrics, and the other one, the column bootstrap, where columns of the expression metrics are bootstrapped. And here at this point it is important to note that the data I will present you in a few moments are all generated from replicate 1, and we can use these results, the results for all methods, and compare them with replicate 2, giving us an idea how close the artificial samples are with the data that was generated in the lab. For this figure, we use the p-values of all detected genes. After doing a differential expression analysis, we calculated the pairwise p-value distances with Spearman's rank correlation coefficient ρ. And we can see when comparing p-values of detected genes that bootstrapping reads seem to be more consistent when regarding variability. So this is this part down here. So FP stands for FastQ bootstrap, and we can see that it is closely clustered together with both true technical replicates, so R1 and R2. And we also redid this analysis when generating artificial samples from replicate 2 compared to replicate 1, so the other way around. And we also looked at the log2 fold changes with similar results to this one presented here. Next, we can take a closer look at the detected genes. We filtered the results for significant differentially expressed genes. In this case, a gene is considered differentially expressed if its adjusted p-value is less than 0.05 and its absolute log2 fold change is greater than 2. And afterwards, we looked at overlapping genes in all methods, and we can see here that we did, first of all, 10 repetitions for all methods, which can be seen here. So FP stands for FastQ bootstrap, CB for column bootstrap, and MO for mixing observation. And when looking at the first row in the heat map, we can observe that all methods had a strong overlap of genes with the results from the first replicate where the data is generated from. However, if we look at the second row, The overlaps with the second, with the true replicate 2, so R2, we can observe that bootstrapping from reads have a little stronger overlap. And also we did a gene enrichment analysis, and we visualize it as the same way as this heat map here, and we could observe the same trend. So we are still working on using these findings to make RNA-seq experiments a little bit more robust, but these preliminary results suggest that bootstrapping reads could help to increase the power of differentially expressed gene analysis. And with that, I thank you very much and wish you a nice evening. Goodbye.